welcome everyone. Today we have James Bradley joining us. I got to know James through his work on cryospheric systems. So he's been looking at glacial forefields and permafrost and glaciers, looking at the geobiology, biogeochemistry, and the evolutionary dimension within those systems. But James is just as comfortable and extremely knowledgeable about deep ocean environment systems too, and I have a feeling that's what we're going to be speaking about today. And for me, the unique selling point of James is that he brings a really nice modeling approach to many of the systems that he works on. And as a result, he's produced some really lovely pieces of work. He's a great asset to the field, and I'd encourage everyone to try and collaborate with James and bring his expertise in that side of things into their work too. So he's currently working as a lecturer at Queen Mary University in London. And since his PhD, which he did at the University of Bristol, James has held 2 fellowship positions. One was at the Deep Carbon Observatory at the University of Southern California. And then more recently, James was actually awarded a Humboldt Fellowship, and he was working at Potsdam. I will leave it there. As always, the format is that James is going to give a seminar, and then we really welcome and encourage as much interaction as possible. So do unmute yourself at the end and ask questions, turn on your video if you want to engage. And of course, if you'd rather not do either of those, you can also write in the chat and I can try and give your questions to James for you. Yeah, so take it away, James. Over to you.
Thanks very much, Karen. Thanks so much for the really kind introduction, and thanks to all of the organizers for inviting me to present my research at this awesome seminar series. I've really enjoyed all of the talks so far. It's been a hugely valuable resource, and I'm looking forward to all of the seminars coming up over the next few weeks. So I'm going to be discussing, as Karen said, the nature of microbial life, microorganisms, and their role in geochemical cycling in the sediments that underlie the ocean floor. So we're going down to the bottom of the ocean and then down even further, tens of meters, hundreds of meters, even kilometers into subseafloor sediments, where we find some of the strangest organisms that are alive, or debatably, as we'll see within this talk, alive on this planet. And they're really interesting because they're perhaps some of the oldest known organisms on Earth in terms of their lifespan, the longest living organisms. They live under extreme conditions for a very long time and give us potentially some analogs to the potential for life elsewhere in the universe. So I'm going to be discussing this habitat, and the theme of the talk is sort of the interaction of these organisms with energy that is kind of fundamental to everything that life as a whole does. So I'm going to be talking about life deep within the Earth. If we want to think about sort of characterizing a biome, or characterizing an environment on the surface of the Earth, this is fairly straightforward. There are obviously complexities and nuances to it, but we can take direct measurements. We can go out and take samples. We can even use we can get a more regional or even a global picture of the nature of life on the surface of Earth by, for instance, satellites. So data that we're seeing here showing us, some of the global terrestrial and marine biomes. But if we want to go into the subsurface, our access and therefore our understanding of the subsurface habitat is much more limited to, for example, opportunistic access via mine shafts or specific holes that have been dug into the terrestrial biosphere or into the deep marine subsurface in order to get sort of snapshots or windows into the nature of this environment. But these are sort of very distributed throughout Earth, and there's you know fairly poor coverage of windows into the deep subsurface and the nature of what it's like to live in the deep subsurface. So hopefully throughout this talk, I'll be kind of filling in some of this black space using mathematical numerical techniques. So within the subsurface, within the deep subsurface, there is surprisingly life, and this is really surprising. It was never thought that there was that the subsurface, especially the marine subsurface, would have been a viable microbial habitat until the early 90s. Since the marine subsurface, when you go beneath the seafloor, it's cut off from light. There's no light there. It's a dark environment. There are no fresh sort of deliveries of food, if you will, of organic matter to feed these organisms for perhaps tens of thousands to millions of years as sediments continue to be sort of advected through, buried into deeper into this environment. And in a lot of cases, there's no oxygen in these environments. So it's a really foreign environment, really different to kind of biospheres that we know harbor the majority of, or harbor life that we're most familiar with on Earth. But when a group of scientists went out on a drill ship, similar to the one that's in this picture at the top, you can see a large drilling rig on the top of this ship, specifically designed to be able to drill down into the marine subsurface. And they recovered sediment cores. So sediment cores extending, this is in meters below seafloor, from like 10 centimeters deep all the way to sort of 1,000, up to 1,000 meters in depth below the seafloor, big long sediment cores. And they examined those cores, and they found evidence of intact microbial cells on the order of sort of hundreds or tens of thousands, up to even millions of cells per centimeter of cubed of mud that they recovered from this environment. And this was a really surprising finding at the time, because it's such a, as I've said, such a foreign, harsh environment to be harboring microorganisms. Some more recent work taking data from subseafloor sediment cores that have been drilled all across the globe and upscaling that to give a global estimate of the number of microbial cells that live in subseafloor sediments. So how many microbial cells are contained in all of the world's subseafloor sediments? Well, it's around 10 to the 29 cells, which is roughly equivalent to the total number of cells in all of the world's soils or all of the world's oceans. But as I was saying in the previous slide, with our global map of the surface and the subsurface world, our understanding of this subsurface biosphere is much more limited than, for example, our understanding arguably of microbes that live in soils, even though this is an equivalent, this biosphere is equivalent in size and is important in a number of different and really interesting ways. One thing that we do know about the nature of life in the subsurface is that it's active. So these cores that I was talking about, you can see in this photo scientists from the drilling ship carrying a core of sediment that has recently been dug, and they're all wearing gas masks. And the reason why they're wearing gas masks is to protect themselves from the, in this case, hydrogen sulfide gas that's given off as a byproduct of microbial metabolisms within the core. So we know that these organisms are active. There's a sort of an outstanding question of how active, and we think that it's extremely low compared to most of life that we're familiar with. We know that these organisms are incredibly persistent, so we can drill down into sediments incredibly deep, and the deeper down you go into sediments, generally the further back in time you go, because sediments in the ocean are deposited kind of sequentially, and as you have more sediment buried on top, your layers get buried in a sort of lower in the profile relative to sediment that has more recently been buried on top. And so scientists have previously drilled down into deep ancient sediment, for example, these subseafloor coal beds from Fumio Inagaki's work in 2015, and have shown that there are microbes that remain active even after sort of tens of thousands, millions of years of burial in these extreme environments. So these microorganisms are incredibly persistent, and they're incredibly important as well for controlling the amount of carbon that is buried in subseafloor sediments. So arguably, marine sediments are the only sort of long-term geological, long-term carbon sink on a geological scale, well, considering kind of sequestration into rocks as well. But organic carbon that's buried in marine sediments that survives microbial degradation is generally buried for timescales of millions of years. And so the activity or inactivity of these organisms has a large bearing on how much of that organic carbon is buried and sequestered into the long-term carbon cycle versus is degraded back into inorganic carbon and goes kind of back into the ocean and back into the faster carbon cycle. Okay, so returning to this compilation of marine sediment cores that was made by Jens Kollmeier and published in 2012, this global database. They did some really nice work characterizing also the number of microorganisms from these really microbial deplete or replete sediments. But here we can see this global compilation of the concentration of microbial cells recovered in sediment from only tens of cells per centimeter cubed to hundreds of millions of cells per centimeter cubed, and a trend as you go down in depth. So this is in depth below meters below seafloor, again from like a log scale, so from kind of just a few centimeters below the seafloor to upwards of a kilometer beneath the seafloor. And a couple of different trends emerge. You can see this for each site. There's a kind of decline in biomass, in cellular biomass, as you go deeper and then into older sediments. And then also depending on where you drill, you find kind of different numbers of cells. This relationship is somewhat sort of has a geographical aspect to it. The colors are the different geographical locations. And if you were to translate, as I was saying, this depth below seafloor into the time since burial, because as you go deeper into sediments, you're going back in time, you see another kind of trend emerge, which is a declining cell biomass from tens of millions of cells in recently deposited sediment that's only 100 or so years old to sediment that's tens of millions of years old. But these sediments are containing extremely few cells, so only sort of 100 cells per centimeter cubed, which is kind of the equivalent of finding 100 golf balls in a stadium full of mud. So it gets really difficult to even find and count these cells when you get down to sort of this level of biomass within the system. But we still have microbial cells that remain somewhat intact in this extremely old sediment that's tens of millions of years old. So the suggestion would be that because they are still intact cells, they've still got to be using some amount of energy to remain intact in order to sort of maintain their cellular membrane and carry out kind of minimal amounts of repair. And they need energy in order to do this. So all life on Earth requires energy in order to stay alive, in order to grow, or in order to just kind of maintain, in order to maintain their cellular membranes, as I was just talking about. And all life on Earth gets this energy through the catalysis of reactions that are exergonic. So reactions, exergonic means energy yielding. And they use this energy to fuel endergonic reactions. Endergonic reactions, those reactions that require energy. So for example, to build a new cell, if the cell is growing, it needs to build all of the biomolecules to produce a new cell, and then it will divide, and then you have two cells. Or similarly, if a microbe is doing maintenance and it needs to repair some of its biomolecules, it needs to synthesize those biomolecules, which costs energy. Really an important thing to get your head around when we think about microbial metabolism and their relationship with energy is this notion of power. So power is energy per time. It's essentially your energy flux. So power typically measured, for example, in watts, a unit that you're familiar with from a TV or a light bulb or a hairdryer. When we think of something being like 20 watts, that means 20 joules of energy per second. So microbes are using a particular amount of energy per unit time. And if we talk about microbes being active and dormant, an active microbe would be expected therefore to be at a higher power, using more energy per time than a dormant microbe. And within marine sediments, we think that the primary electron donor within this redox couple is organic carbon. And therefore, if we plot the organic carbon content of the sediment cores where these cells were collected from, we can see another trend emerge, which is in the relatively recently deposited sediment, we have high concentrations of organic carbon, and in the older sediment, we have lower concentrations of organic carbon. And so the sort of suggestion there would be that microbes are using this organic carbon to fuel their microbial metabolism, their metabolisms. So energy is important for a whole range of different things. Energy is needed for microbes to grow. And if microbes are growing, they're carrying out cellular division, they are therefore evolving as well in the kind of traditional use of the word evolution is accumulation of mutations through cellular division. So if we have growth, then we have evolution. We also then have the potential for microbial communities to assemble according to specific kind of gradients. But if there's not enough energy for this growth to happen, maybe we can't have this sort of assembly and evolutionary mechanism. We still don't really know what the least amount of energy is to sustain microbial life. So if we're thinking about looking for life on other planets, if we can characterize how much energy is available, and then we have an idea of the rate at which microbes use that energy, we could potentially figure out how long microbes can be sustained within this habitat. So it's a pretty important thing to figure out if we want to characterize whether or not an environment might be conducive to harboring life. And as I said, microbial metabolism is very important for the global carbon cycle. So whether or not microbes have enough energy to live and to grow will have a bearing on how much organic carbon is buried within sediments and sequestered versus is put back into the fast carbon cycle and back into the atmosphere. So we want to understand microbial energetics. What's sustaining these cells? There's a general decrease in cell number as we go down into the more ancient sediments, which means that this system is essentially kind of a net death system. And as you can see here with these colors, there's very little organic carbon left in these sediments by the time you get into the deeper ones. So an outstanding question maybe is, since there's so much microbial mortality, since there's so many dead cells being produced, we've got tens of millions of dead cells, does that provide a sorry, my computer something popped up there. You can still see the presentation, right? Yeah. Does that provide, do all these dead cells provide an opportunity for the survivors? And we know through various lab experiments that actually, yes, dead cells do provide a significant source of energy to the surviving cells. So if you sustain a culture of cells in a lab under very low energy conditions, they enter into this really interesting kind of long-term stationary phase called the gasp response. And the survivors at least start using relic DNA as a nutrient. They start kind of cannibalizing themselves, and you get these really interesting dynamics. So maybe we could have something similar in the subsurface where you have this net death, a large production of necromass, and that sustains microbial life. And this is kind of one question that I went after, but unfortunately we can't do these experiments in a lab, because even though you can starve a culture of cells in a lab, they were once recently growing, and you can't really replicate sort of 10 million years of burial in sediments within a lab environment. So we took a new approach, which was to use modeling, and specifically bioenergetic modeling and calculations of energy and power, try to answer this question. So, what time is it? I'm going to have to go a bit quicker, I think, to get through this stuff. But essentially, what we want to do is compare the power demand of microbial cells that are within the subsurface environments and compare that with the power supply. So the power supply can be calculated by the Gibbs energy of a reaction, and in this case, the rate at which that reaction is occurring, which is the rate of necromass production, the rate of the production of dead cells within our environment. And we can calculate what the supply of energy is from these dead cells. We did this focusing specifically on an environment from the South Pacific Gyre, which is an area of the ocean which is very low in sea surface chlorophyll. So there's extremely low productivity, which means that we think the microbes that are living in the sediments beneath this environment are surviving on very low energy fluxes. So we kind of tested this bioenergetic model there. So we looked at how many microbial cells there were within the sediment, calculated based on the decline in cell number, how many dead cells it produced, and then could calculate a cell death rate from that. And these are extremely low cell mortality rates, the order of sort of 10 to the -6, 10 to the -7 per year. This is sort of, if you were to compare these mortality rates to the mortality rate of microbes within soils, it's at least like 5 orders of magnitude lower. So already we're getting kind of clues that this environment is very different to one that we're more familiar with working in surface environments. So what is necromass? Necromass is a dead cell. So we can split down a microbe into all of its biomolecular constituents, the amino acids, the nucleotides, and use free energy calculations to estimate how much energy is produced when you were to oxidize these biomolecules using an electron acceptor, such as, for example, oxygen or sulfates, depending on the environment that we're in and we want to calculate. And so we do these free energy calculations. I'm going to go through this a little quicker. But essentially what we get is power available to microbes from the oxidation of necromass that is being produced in situ, the demand from the microbial community, massive error uncertainty on here because there's very few suggestions in the literature of how much energy or how much power these microbes demand. But actually, since even within our sort of 4 orders of magnitude uncertainty, it falls to the left, or necromass energy available from necromass production falls to the left, or lower than the demand of the microbial community. And the amount of energy available from organic carbon degradation, which we also calculated, falls sort of within this range. So this is just a suggestion, therefore, that the amount of energy available from microbial necromass production in the marine subsurface is not sufficient to sustain the demand of the microbial community, and likely the microbes are using organic carbon as their primary energy source, so organic carbon that is deposited already within the sediment. It's interesting to think about how to represent microbial metabolisms within process-based models. And so I've done a little modeling on soil microbes and a few other environments. And traditionally, the equations that you use to simulate microbial growth is you have a pool that represents how much biomass you have, and you have a pool that represents how much organic carbon that you have, and then the microbes consume that organic carbon and they grow from that. There's more simplistic models that don't resolve microbes at all, and then you just have this kind of a parameter that represents the rate of organic carbon degradation and transformation into CO2. But these models don't really work for the deep subsurface, where you don't really have much, or we don't think there's much microbial growth. So we still have organic matter degradation, but we don't have maybe any microbial growth. So what we need is a term for maintenance, a term for the consumption of organic carbon due to not just microbial growth represented by the black line, but also the consumption of substrate due to microbial maintenance. So we have 2 options for how microbes can use energy for maintenance. Either they use their own biomass or they use organic carbon substrates. And typically, if substrate is limiting, microbes will start to metabolize themselves as a sort of survival mode almost, or reduce the amount of energy that they're using by going dormant. So we also need to represent dormant microbes. We have a number of different ways of doing this within models. One would be to have a kind of active fraction, a parameter that represents our proportion of active microbes versus dormant microbes. And another would be to have discrete pools, a pool representing active microbes and a pool representing dormant microbes, with kind of activation and deactivation occurring depending on the prevailing environmental conditions and maybe the amount of substrate available for their growth. So I kind of wrap these two things, the maintenance and the dormancy, together with a new sort of combined approach where microbes can switch between active and dormant states, and they can use organic carbon for growth, and they can also use organic carbon for maintenance energy, but they can also use their own biomass for maintenance energy. And very briefly, again, because I don't have time to go into details, but the citations are given here if you want to read about it in more detail, develop this kind of new physiological model where microbes are interacting with the substrates, and they're going through these phases of being super active to being super dormant, and their mortality rate and their maintenance energy demand kind of changes depending on which physiological state the microbes are in. And we learned a few things by applying this to marine sediment environments. We learned that based on the amount of energy that's available and the amount of energy that microbes are using, there isn't really much of an opportunity for new biomass growth. Microbes will go pretty dormant pretty quickly if they're going to survive within this environment. So in order to explain there being preservation of microbes still within deep ancient sediment, you need for there to be not much growth. You also need for there to be organic carbon consumption that's dominated by catabolism to support maintenance activities, which kind of goes hand in hand with the first one. Organic carbon is not going towards growth, biomass synthesis for new cells, for cell division. It's going towards maintenance. And when we look at profiles of organic carbon within marine sediments, you can see that they decline very steeply. So we're getting a lot of organic carbon consumption due to maintenance activities. So therefore, we're having just really quite extremely dormant microbes having a very considerable impact on the global flux of organic carbon through marine sediments. So this is kind of, we're learning that you don't necessarily need super active microbes to have a large bearing on global biogeochemical cycles. You can have a lot of dormant microbes over a very long time having a really significant bearing on the fluxes of carbon and other elements through the Earth system. Okay, thinking about energy in subseafloor environments and thinking about moving from location to location, we can try and quantify how much energy these microbes use. So here I've got a human, and here I've got a microbe from the deep subsurface. A human, surprisingly, I think, uses about 100 watts of power, which is kind of the equivalent of like 2 light bulbs or a powerful hairdryer, which, you know, I think that seems quite low, but that's what we run on. A microbe from the deep subsurface runs on much less than that. A microbe from the deep subsurface runs on, and I always get this wrong, like 100,000 million billionth of the power of what a human runs on. So we're talking in the order of zeptowatts, and zeptowatts is zepto means 10 to the minus 21. So it's a unit, you know, that is, I'm now very familiar with, but we don't tend to use much when we're talking about these other systems. So microbes from the deep subsurface are very different to our kind of understanding of the majority of life on the planet. But we only have these estimates from a few distinct geographical locations. As I said, like, our access to the marine subsurface is limited, and we have limited data and limited measurements, and a whole lot of the marine subsurface is a kind of blank box that we haven't yet filled in. So I intended to fill in the blanks basically to try to quantify how much energy per time microbes from the subseafloor are using on a global scale, because it's important for all of these different questions that we talked about before, whether microbes can grow, whether or not you even have evolution within this system that comprises the same number of cells as there are in all of the world's soils or all of the world's oceans. So I set out to do this. The only way to really get at that was to use a modeling approach. And the overall kind of framework is fairly simple to get your head around. There are nuances in how it's implemented, but we want to quantify the power per cell, the rate of energy use per microbial cell. And in order to do that, we need to know what the rate of reaction is. So this is our time term, the delta G of that reaction. So this is providing the energy. So we'll get joules here, and the rate is our per second term. And then how many microbes are in that environment? We divide by how much that energy is shared by the number of cells, and we get power per cell. So we need to do this for all of the marine subsurface. So myself and colleagues divided the globe into lots of grid boxes, quarter of a degree by quarter of a degree grid boxes, which gives us, several 100,000 grid cells. And we used various modeling approaches to calculate how much organic carbon there is at specific layers and specific ages of marine sediment within these grid boxes, and between layers and between ages, based on a whole load of kind of global data sets and global parameters. And this gives us our rate of reaction. It gives us the rate at which organic carbon is being degraded. I'm happy to go into details about this, but maybe not right now. Then we need to know the delta G term, the free energy that's available from the rate of organic carbon degradation. So we need to know what the sort of major catabolic strategy of these organisms are. Are they oxidizing organic carbon using oxygen, or are they using sulfate, or are they carrying out methanogenesis, for instance? So for every grid box, we need to know where is oxygen, how deep does oxygen penetrate, where is sulfate available and how deep does sulfate penetrate, and then beneath that we have our kind of methanogenic zone, and then beneath that we have the basement rock. And what we did is we used some global data sets that enable us to predict the prevalence of oxygen and sulfate through global marine sediments in order to sort of map those transition zones onto our global framework. And yeah, there are some. So this is a simplification of how of microbial metabolisms, and it is we're not considering, for example, nitrification, fermentation. So there is potentially some misattribution of energy between these different groups, but work that we've done and work that others have done have shown that these metabolisms capture the majority of energy that's available, at least in sort of the realms in which we're considering. So the first few million years of burial and the majority of microbes in marine sediments. Some really interesting work from Justine Sauvage and Steve Dont and others have shown that radiolysis provides energy to microbes in marine sediments, which is it's a super cool study that was out a few weeks ago in Nature Communications. Their finding sort of aligns with ours in that organic carbon is the major source of energy, and that after several millions of years, organic carbon provides about 98% of the energy available, and then radiolysis of water provides around 2% of that. But they're really interesting sort of take home from their paper was that the radiolysis of water is a kind of infinite source of energy. So that's potentially then available to sustain microbes when even organic carbon sources have been exhausted. But anyway, I've got distracted there. I'll keep on with where I'm going. So we're looking at the free energy term. We can calculate how much energy is available from the rate of organic matter degradation based on this profile and where these reactants are available on a global basis. And then we need to calculate how many cells are present in our sediment environment for every grid cell. And thankfully, Jens Kollmeier and colleagues have already done this for us in a paper from 2012. So we used their prediction for the prevalence and abundance of cells in marine sediments to use within our global model and make these calculations. Okay, so we're calculating power per cell for every grid cell based on the rate of reaction, the free energy available, and the number of cells. And that enables us to quantify kind of simultaneously the global distribution of electron acceptors within marine sediments. How prevalent is oxygen and whereabouts is it? How prevalent are sulfate reducing environments? How much do they comprise, for example, the global volume of marine sediments? That's what we're showing in this pie chart. So oxic sediments comprise a really small proportion of marine sediments. And this calculation aligns, this is a calculation that aligns really well with some work that Emily Estes has done recently, published in Nature Geosciences, that got at this exact same question through a really different process, so using empirical models versus process-based models. So it's really nice that our findings align on that. We can show that methanogenesis is really prevalent, and it's actually just restricted to these coastal areas. So we can map for any age of sediment and geographical area whether or not marine sediments are oxic, sulfate reducing, methanogenic, for any depth or any age. We can also look at the distribution rates and thermodynamic properties of organic carbon that's stored in marine sediments. So for example, you can see that sulfate reducing sediments store the vast majority of organic carbon that is in marine sediments. Or to put that another way, the vast majority of organic carbon in marine sediments sits within sulfate reducing environments. And also calculate how much is degraded and what is the role of these different electron acceptors in degrading organic matter. So you can see, you know, oxygen is still minor, but it's sort of taking on somewhat of a more major role than its sort of volumetric prevalence. And that is because there is more organic carbon sitting in near surface environments. So it's sort of, and it's a more sort of powerful reactant in a way. So oxygen is slightly more important, but we're still seeing a prevalence of sulfate reduction in the degradation of organic matter on a global scale. And we can also look at how that's spatially distributed. I'm not going to go into the details for that for the sake of time, but I'm happy to return to it. We can use our models to map the global distribution of cells and which environment they sit in. So again, biomass is pretty much tracing organic carbon stored, which is encouraging. That's what you would expect. You would expect environments which contain a lot of organic matter to have a lot of microbes attached to that organic matter. And so the finding that there's not much organic matter in oxic sediments, and there's also not many cells in oxic sediments, those two things kind of align quite well. And again, we can map their distribution across any depth or any age horizon on a global basis. And then finally, sort of the purpose of all of this is that we can calculate the global subseafloor sediment utilization of energy, or over time of power. So this is in gigawatts, and actually you can see that you know, as a global total in gigawatts, oxic sediments, microbes in oxic sediments are using a lot of power, a lot of energy, compared to other sediments. And if we add up the total energy or power utilization of microbial life in global marine sediments, we get about nearly 40 gigawatts of power, which is roughly equivalent to the power output of Denmark, or the power use of Denmark. But compared to, so putting that into a more kind of ecologically relevant framework, the power of microbial life in the global subseafloor is only about 0.1% of that, of the total power of the sort of photosynthetic fixation of inorganic carbon into organic carbon in the surface of the ocean. So real clear distinction between sort of surface dwelling life living on sunlight than deep dwelling life living off of the remnants of that photosynthetic production. So again, we can map this power onto global maps. And now what I'm going to show you is the power per microbial cell in watts. So how many watts of power is an individual microbial cell using if it's situated in oxic sediments? Okay, so the warmer colors is kind of higher on the spectrum. In sulfate reducing sediments, there's still some areas of like relatively higher power than others. And then methanogenic sediments, so we're really at our low end of the power scale. And the reason why there's white areas on these maps is because, you know, in this white area we don't have any methanogenic sediment. Methanogenic sediment's only restricted to the kind of coastal areas. But these powers are incredibly low. We're talking 10 to the minus 21 of power up to 10 to the minus 16. So spanning about 5 orders of magnitude. And even microbes being sustained at the highest power relative to marine sediment environments is extremely low compared to other environments, which is what I'll show now. So here we have this same spectrum of microbial energy utilization in joules per second or watts per microbial cell from 10 to the minus 21 to 10 to the minus 16. We've got our oxic sediments here, our sulfate reducing sediments, and our methanogenic sediments. How does this relate to other microbes that are using energy? Well, we have humans, so about 100 watts of power. That would be sort of all the way further past the end of this slide, another kind of 3 or 4 slides put to the side of this. We have, okay, well, what is, let's just for now ignore all of the microbes and soils and stuff that we're more familiar with, because we want to compare this to like pushing the boundaries of what's the limit to life. So what's the lowest energy flux that we've ever measured in a lab environment? So when you take microbes in a lab and you don't let them grow and they're just doing maintenance and you really starve them, they're using about 10 to the minus 15 watts of power, which plots around here. So all of our subseafloor environments, all of the microbes within it are using substantially less power to survive than where we can push our lab experiments to the limit. You know, still orders of magnitude lower. When we look in natural settings and we measure how much energy can sustain microbes in a natural setting, and this is discounting growth. So this is microbes that aren't growing; they're just doing maintenance, so just barely surviving in any natural setting. We plot about here. So again, all of our microbes in marine sediments are using substantially less or surviving on substantially less energy than has previously been shown for a natural setting. And then using theoretical models and mathematical models, the lowest modeled maintenance power for microbial life in subseafloor sediments plots about here. So, you know, what we found through this work is that about 80% or more of microbial cells in marine sediments, primarily those that are in methanogenic sediments and that are in sulfate reducing sediments, survive on orders of magnitude less power than has ever been shown to sustain life on Earth before. So this is really, to me at least, expanding sort of what we might consider to be an energy limit to life and pushing that much lower. So take home messages from this, and then I'm happy to answer any questions. Hopefully I've left a good amount of time for that. Yeah, hopefully I've kind of expanded this black box of what we know about and where we know about microbial life in marine sediments to somewhat produce these kind of global pictures that tell us about where microbes are, what their energy sources are, and how much energy they're using. We've quantified on a global basis the electron acceptor availability. So where is oxygen, where is sulfate, where is methanogenesis going to be prevalent, and how rapidly are these electron acceptors being used? What's the rate of oxygen consumption? What's the amount of organic carbon that's stored in these environments and degraded in these environments on a global basis? We've just recently shown that the overwhelming majority of life within these marine sediments on a global basis subsists on energy fluxes that are lower than we thought before to even sustain cells. So, you know, all of these microbes are under fairly extreme and widespread energy limitation. And the energy fluxes that we think that they are, you know, surviving or being sustained at do fall quite far short of previous estimates for maintenance. And that, I think that has implications for ruling out almost cell division and biosynthesis growth of microorganisms on a widespread basis in marine sediments. And therefore, this, you know, evolution by sort of the accumulation of mutations due to cell division, I would say that it's likely that that's not prevalent, therefore, in marine sediments on a global basis. We have this whole biosphere that's not necessarily evolving in the way that we think about evolution in that sense. Yeah, and since we've found the life is pretty prevalent on Earth using much less energy than we thought would sustain a cell before. So that has certainly implications for when we look at in other environments on Earth or other environments on other planetary bodies and think about how much energy is available. Maybe this expands the sort of the realm of what we would consider plausible of an environment in order to support microbial life over very long timescales. So I think that's it. Thanks very much for all of your attention, and I'm happy to answer questions.
Thanks so much. Great, James. That was really, really fascinating and so different to all the other talks that we've had in these seminars. So thanks for that. If anyone wants to start off with a question, please shout out or raise your hand. And also, again, you can write in the chat if you want to do it that way. I'll just give a moment for anyone to come forward. Yeah, Joanne, if you want to start us off, thanks.
Yeah, so I assume you can hear me okay, yeah?
Yeah, great.
Okay, cool.
Yeah.
I think I don't know anything about the sea floor sediments, but you haven't talked about autotrophy. You just talked about organic matter being used. Is there a reason for that?
Yeah, so that's a really good question, and it's something that's starting to get a lot more attention in studies of the subseafloor biosphere. I briefly mentioned a recent study from Justin Sauvage, who kind of quantified and characterized how microbial life could be using hydrogen that's produced through the radiolysis of water. So, you know, that process will produce hydrogen and oxygen, and they're two reactants that can provide an enormous amount of energy. You know, that's what's used to send rockets into space. So if microbes can catalyze that energy source, then that could sustain life. They estimated that it comprises about 2% of the energy in subseafloor sediments. So, you know, that becomes important when organic carbon has been exhausted. So when we get into the more deeper ancient sediments, potentially you have microbial life being sustained on that process. There's also various opportunities that arise from the products of organic matter degradation. So for example, you might think about nitrification being one of them, the use of sort of inorganic nutrients and metabolisms that will produce energy. The problem with that as being a widespread source of energy is that the production of these reactants depends on the degradation of organic matter. So in order for there to be the reactants for these various metabolisms, you first have to have organic matter being degraded. And when you when you do the calculations to consider sort of how much reactant might be produced versus how much organic matter is degraded, and compare the amount of energy that's available from that, then organic matter is still the primary source of energy. So these metabolisms certainly are out there.
I mean, you've got iron sulfide.
They just might not be.
Presumably you've got iron sulfide in these sediments. And clearly, if you've got methanogenesis, you've got CO2. So, I mean, there are other electron acceptors. I mean, you've got your sulfate, you've got nitrate, I presume, as well. So I'd like to get a feel for how much autotrophic, you know, how much CO2 fixation is happening. Of course, if you've got organic matter, you're going to have, you know, bugs that will preferentially use that over CO2. But you'll have, I presume, some strict autotrophs in those environments, because you've got a, you know, a lot of different electron donors.
And there is evidence. Yeah, and there is evidence for that within sort of data on gene prevalence and also on gene expression within the deep biosphere. As we kind of currently understand marine sediments, they're not the dominant microbes that are there. And since this is, you know, the purpose of this study, as you know, was to get a handle on sort of the utilization of energy more on a global basis, and it gets, at some point you need to draw the limit of like what you'll include and what you just need to make an assumption for that will kind of be accounted for in other areas. So we don't resolve them explicitly, and trying to resolve explicitly and interpret from data sets sort of what gene is responsible for which function, and then put that into the global model, we wouldn't get anywhere. So we start with this, and then
I
maybe
understand.
we can expand on that.
I understand. If you want to send me some papers to say that autotrophy is less dominant than heterotrophy in these environments, that would be useful. If you've got any papers or any information.
Thanks. Let's move on. Julia, you've got your hand up. We'll go with you, and then there's some in the chat we can go to next.
Yeah, thank you. So that was really interesting. Thanks so much, James. I was wondering if we're fundamentally misunderstanding the ecology of even the shallow biosphere with our lab experiments, because we're subjecting the microbial communities there to a much higher power environment when we do that in the lab. Or do you think the ecology itself is largely preserved? Because essentially, we wouldn't dream of investigating a very high temperature ecosystem by doing experiments at 4 degrees, or it's quite a change in the environment itself if we change the power that is available.
Yeah, certainly. Yeah.
The ecology itself.
So I mean, lab experiments, of course, serve a useful purpose in understanding the physiology of microorganisms, and they're absolutely essential to gaining all of the knowledge that we have about how microbes react to different conditions and the ways in which they use all of the food sources available to them in the products and the pathways and all sorts of things. But how now, relatively speaking, high energy lab environments and experiments are to the deep biosphere.
I mean, even the shallow biosphere.
Yeah, there is a.
To be honest.
Right.
Because.
Yeah.
I mean, we can't really. I guess we're not even trying to replicate the deep biosphere so much in the lab, because we humbly know that we can't. But even for the shallow.
And I think. Well, actually, this shows that quite well. I think that. Yeah, if your intention is to replicate the deep biosphere in a lab environment, that's going to be really challenging, right?
Yeah, absolutely.
You can't even represent 10,000 to 1,000 years of burial, which is a very short time scale, thinking on the order of the deep biosphere within a lab. But you don't need to go very, as you were saying, our shallow biosphere, you don't need to go very deep to get microbes that are subsisting at extremely low powers. So our methanogens are subsisting at the lowest power, and methanogenesis is restricted to sort of coastal environments, and you can find methanogenic sediments in relatively shallow environments close to the coast, where you know, you don't need to go right into the middle of the oceans to find these extreme energy limited zones. You can go just off the coast a few meters deep into the sediment, which represents a lot longer, sometimes longer timescales of burial than you know, other shelf environments or other sort of margin environments.
Mm-hmm. Yeah, no, I just think I hope we're just not misleading ourselves by thinking that we actually get a good grasp of the, you know, the more accessible environments with our lab experience. When in fact, you know, everything is a very different context, I guess. But okay, yeah, thank you.
So we've got loads of great questions in the chat. Let's see if we can get through some of those. Ashish was asking if deep sea sediment microbes are dormant, what maintaining is there to do? Just thinking out loud. Thanks, Ashish. So yeah, what maintenance are they doing?
What are they doing? Yeah, well, I mean, that kind of depends on how you classify active and dormant cells, and to me at least, dormancy still requires some basal amount of metabolism. One, you need to still remain an intact cell, so you need to fight the pressures of your external environment to not be just turned into your external environment. And two, you have to be able to sense the outside environment in order to know when is a good time to wake up and be active. And both of those things require energy. So even though we have sort of active cells and dormant cells, and then even more dormant cells, and then maybe even more dormant cells, that in order to be counted as still alive, I think these cells are still using some amount of energy. And again, then it questions your definition of what is being alive. Do you need to be able to be resuscitated in order to be classified as alive? Well, not if you're just continuing to use very low fluxes of energy to maintain your state. Certainly not all dormant cells will be resuscitatable, but they might continue to be metabolizing in this dormant state. It's a really, it's a tough question, because I think maybe our way of thinking about this is almost biased by like how we're familiar with most surface life, and these microbes just operate on very different timescales that we can't really understand.
Yeah. And kind of carrying on from that, James is asking about the minimum level of power output, thinking about like Mars and
so how much power is available?
Like what would be the lowest level that you could,
if you could quantify rates of geochemical reactions in any environment, that gives you information about how much energy might be available to the microbes. Similarly, if you have like a standing stock of reactants, you could calculate based on the power requirement of life, how long those reactants might sustain life if they were to be used to a full extent or half, or you know. So if you could have data, for example, from the surface of Mars or the subsurface of Mars on reactants and products and reaction rates, you could make some calculations as to how many microbes might be sustained for how long.
Could I just throw in something to spin James's question just a moment, because I actually had a similar question, and sorry, the other James, but now this James. You talked about a gap between sort of the surface energies and this subsurface ocean seafloor energies, like you said, it's 0.1 or something like that of the available energies near the surface. Along those lines of what you were just saying, I guess we could sort of predict what types of energies might be available from mineral assemblages or predicted mineral assemblages in the subsurface of someplace like Mars, right? How can you comment as to whether you could compare the sort of delta energy available between, you know, superficial geochemical reactions and subsurface reactions in a place like Mars with the type of energy difference that you were talking about? And is it potentially within that sort of range?
So I can't provide any specific numbers on that. I haven't thought in enough depth about that, but it might well be that there is an abundance of energy available, for example, on the surface of Mars, but that other conditions make it not conducive to there being life there. So, that there's a niche beneath the surface, perhaps, where the energy availability might be much lower, but you might have much less ionizing radiation and much, more suitable like conditions to sustain life over a longer term. So energy is important. It's certainly like a primary factor on microbial life as a whole, but there are other factors that we of course need to consider that impact mortality and
of course
survival. And so I can't give you any specifics on that, but that's a great question.
I wasn't asking for any specifics. That's okay. But thanks, that's really fascinating. Thanks.
We are just past 1 o'clock. Are you okay to stay a little bit, James? There's a couple more questions we could maybe try and tackle. And just for anyone that needs to go, we've got another talk in 2 weeks' time, Catherine Duncan, and I've left the title in the slide, in the chat. There's a couple on kind of, have you been able to sequence these guys? Do we know who's down there?
Yeah, not me specifically, but certainly there are a huge number of publications that have got genomic data from these subseafloor environments. Mm-hmm. And yeah, there's an abundance of that in the literature. Yeah.
And then Richard is asking, is the DNA purer and simpler as the cells have not evolved so much and does not give a window into the origin of life?
Right. So if yeah, I mean, there's a nice kind of, I don't have the specific answer against that, but there's a nice kind of conceptual way of thinking about that question, which is if you presume that there hasn't been evolution for 50 million years between microbes that have been deposited in this deep sediment, can you see that they're evolutionarily distinct from microbes that are in these surface environments in the same location? And there's been attempts to look for kind of tracking rates of evolution through microbes across these long and deep ancient sediment profiles, but there's issues of like changing surface environments throughout this time, changing geochemical conditions, being able to distinguish what is a difference arising from evolution versus anything else among taxa that might be minor fractions of your entire community. So it's a really interesting kind of thought experiment, but putting into practice is kind of plagued with all of these difficulties.
Yeah. Speaking about difficulties, have you got any sense on the margin of error in calculating like power consumption?
Yeah, so we made attempts to quantify sort of what errors might creep in from various components of the reaction framework. So if you remember, we had power per cell equals rate of reaction multiplied by delta G divided by the cell number. And there are certainly uncertainties in all of those terms and the parameters that feed into those terms. So to quantify uncertainty on the rate of organic matter degradation, we looked at sort of the entire plausible range of parameter sets for every grid cell and calculated a kind of min-max scenario. To look at uncertainties for the delta G, you can consider different concentrations of reactants and products and potential error in how we assign metabolisms to the different zones. And then for error in the cell abundance, okay, well, you can also consider what if only 50% of those microbes were carrying out the metabolism that was ascribed to it. And we did quantify potential error in all of those things. And you do get some fairly considerable variations. So, this can you see my mouse? These peaks might move, 10%, 15%, which in an ordinary study, where you're not dealing with this order of magnitude range, might be considerable. But 10, 15, 20% error and movement of these peaks doesn't change the fact that they're still falling far beneath, what we would have previously considered to be a power limit to life. So the error can be is certainly there, but we think that it's not considerable enough. You know, you would need to have orders of magnitude of error to be able to move these peaks to change the story. So we're fairly confident in the.
I guess it's also complicated because you've pulled down data sets from other studies, like the cell abundance. I think you'd taken from other folks, so it must all get very complicated to deal with.
Yeah, and so we think about, you know, what if we're wrong by a factor of 2, and what are the implications of that? Yeah.
Let me interrogate you with one last question. Very interesting to see insights into the biogeography of power usage. Do your models of energy availability used to make predictions about the promotion of active versus dormant cells? And if so, has it been possible to factor this into your estimates of cell-specific power consumption, as this presumably introduces a bimodal distribution caused by lower use by dormant cells?
Yeah, okay, so yeah, one thing to do would be to align the kind of process-based modeling of the active and dormant cells and the fluxes between them with this bioenergetic modeling, and thinking about, okay, well, if we have this amount of energy in this environment, is that leading to an exchange of microbes between this active dormant state? It's something that I haven't done on a global basis. It certainly could be done, and it would probably be quite valuable to do that. On the sort of bimodal distribution, or these, you know, you can see this peak here has an extra peak here, so they are somewhat they're actually kind of trimodal. There are 3 peaks in there, so afterwards there's a almost indistinguishable peak here, and that's actually due to whether or not the sediments, in this case methanogenic sediments, are in a shelf zone or a margin zone or in the abyssal zone of the ocean. And actually you see quite different phenomena when you compare coastal margins to shelf environments to the abyssal deep zones of the ocean. So that's what's causing these kind of bimodal distributions.
Brilliant. Lots of praise of your talk in the chat, and some good comments. Zito, what is the word of the day? Apparently, so right. I think we should let you off the hook now. You've been brilliant, James. Thanks so much for the talk, and
yeah, thanks for having me.
Yeah, thanks everyone else for joining us, and for any of you having Easter breaks coming up soon, I hope you have a wonderful time and very relaxing and peaceful.